back to another episode of Bacteria Live with your host, Dr. Phage.
Thank you everyone. As you know, today we'll be discussing some of our most exciting topics. Transformation, transduction, and bacterial sex, aka conjugation. Well, let's jump right in.
Don't go changing to try and please me.
Hi guys, I'm Bacteria Bob. Today, we're going to talk about transformations. Transformations involve the transfer of naked DNA into a recipient cell, changing their genotype and phenotype.
Now let's go to the cartoon.
Step 1. First, double-stranded donor DNA binds to specific receptors on the surface of a competent bacteria cell. Step 2. One strand of the donor DNA is degraded by nucleases while the other DNA enters the cell. Step 3. Single-stranded donor DNA pairs with a homologous region on the recipient DNA and is integrated into the recipient genome by a breakage and reunion mechanism called homologous recombination. This also occurs in the opposite strand of DNA. Step 4 of transformation. Following homologous recombination, bacterial cells asexually divide, resulting in daughter cells that contain the new genetic information.
I wanna love you and treat you right. I wanna love you.
And now for a lesson about transduction with Dr. E. coli.
That would be me. Hello, I am Dr. E. coli. And today I would like to tell you a story about transduction and a little lonely bacterial cell. Here it is now. This little lonely bacterial cell wondered if it will ever find the one. Instead of obeying its parents' wishes, it yearned for a bacterial phage. Suddenly, its life is forever changed when that virus asked it to get cozy. One thing leads to another, yada yada yada, and the bacteria now has viral DNA.
Step 2 of transduction.
Break it down now.
Following the addition of viral DNA, a phage enzyme is produced that breaks down the host DNA into smaller fragments. Phage DNA is replicated and phage-coat proteins are produced. Now for step 3. During formation of the mature phage particles, a few phage heads may surround fragments of bacterial DNA instead of phage DNA. The phage particle then carrying the bacterial DNA infects the another cell, transferring the bacterial DNA to the new cell. Finally, step 5. When the bacterial DNA is introduced into the new host cell, it can become integrated into the bacterial chromosome, thereby transferring genes to the recipient. This cell multiplies and carries new genetic material to 2 new cells.
I believe in miracles. Where you from? You sexy thing, you. I believe in miracles. Since you came along, you sexy thing.
Conjugation is a mechanism of gene transfer that requires direct contact between donor and recipient cells.
F+ cells carry the F plasmid, enabling it to make an F pelus and act as a genetic donor. Recipient cells that lack an F plasmid are referred to as F-.
The F plasmid encodes for the F pelus, a protein appendage that attaches the donor to the recipient cell. The first step in plasmid transfer is contact between the donor and the recipient.
The F pelus of the donor cell recognizes and binds to specific receptor sites on the cell wall of the recipient cell.
The plasmid then becomes mobilized for transfer when a plasmid-encoded endonuclease cleaves one strand of the plasmid at a specific nucleotide sequence called the origin of transfer.
A single strand of the F plasmid beginning at the origin of transfer enters the F- cell.
A complementary strand to the single-stranded plasmid remaining in the donor is synthesized via the rolling circle mechanism.
Once inside the recipient cell, a complementary strand to the single-stranded DNA is synthesized.
Now we're one of a kind!
When F plus and F- cells are mixed together, eventually all the cells become F plus.
And the two bacteria cells lived happily ever after.
My darling, I can't get enough of your love, babe. Girl, I don't know why. Can't get enough of your love, babe. Lord, some things I can't get used to, no matter how I try.